Our final speaker is Sherrianne Ng, a research associate in bioinformatics in the Department of Metabolism, Digestion, and Reproduction at Imperial College London. Sherrianne received a PhD in biomedical science at Murdoch University in Perth, Australia, studying sepsis in preterm infants. Today, we'll hear about new work on specific maternal factors that play a role in preterm birth risk and sepsis.
Thank you to the organizers for letting me present my research, which is on insights into preterm birth using multi-omics approaches. So why is preterm birth important? Preterm birth affects 15 million babies every year, accounts for more than 1 million deaths globally, and is a substantial economic burden on the healthcare system. Preterm birth is very complex to solve. There are a range of factors that can cause preterm birth, from smoking to ascending infections, or the cause could be unknown. Babies who are born preterm are also at higher risk of developing infections as well as other long-term morbidities. So how can we understand preterm birth better? Increasing studies show the potential of multi-omics approaches to capture the complexity associated with preterm birth. There are a range of omics fields, from genomics to metabolomics. These can give us different levels of insight into how different biological systems are contributing to the phenotype, which is the physical trait we see happening. Today, I will share two of my research projects, which focuses on understanding preterm birth from a microbiological perspective in both the mother and the baby. The first uses multi-omics approaches to improve our understanding of bacterial bloodstream infections, also known as neonatal sepsis, in preterm infants. Neonatal sepsis accounts for 15% of deaths within the first month of life, causing significant mortality and morbidity, especially in preterm infants. There are serious implications due to the lack of early detection, prompt diagnosis, and appropriate treatment of sepsis, as well as limited understanding of early life immune-metabolic responses. Our study combined the use of both transcriptomics and metabolomics to identify blood biomarkers that is distinctly present during sepsis in preterm infants born less than 30 weeks gestation. Following patient recruitment and sampling, we processed whole blood for transcriptomic RNA-seq analysis, metabolomic liquid chromatography mass spectrometry analysis, and integrated different data types for patient classification. Based on overall gene expression, the transcriptome of septic infants, the ones in red, appeared to be more similar to each other, but different from infants with possible or no sepsis, the ones in green and blue. More importantly, the blood leukocyte transcriptional responses of septic infants was significantly altered compared to infants with no sepsis, with 1,317 differentially expressed genes. Based on network analysis and pathway overrepresentation analyses, we identified that these transcriptional aberrations in host responses during confirmed sepsis were associated with four main functions. First, that pathogen and pattern recognition mainly function through toll-like receptor pathways, which is not surprising, considering these receptors recognize a range of bacterial components from both gram-positive and gram-negative bacteria that are often implicated during sepsis. Second, for cytokine signaling, we found that both pro-inflammatory cytokine signaling and immune inhibitory signaling were linked to upregulated genes. These were mainly your interferon and interleukin signaling genes, as well as immune inhibitory signaling genes such as IL-1R2 and IL-1RN, which inhibits interleukin-1 signaling. Third, that various pathways associated with immune and hematological regulation were overrepresented. More importantly, that despite most genes being upregulated, two key genes important for regulating apoptosis, BCL2 and IL-7R, were downregulated, suggesting that genes involved in apoptosis are likely dysregulated during confirmed sepsis. So the importance of crosstalk between metabolism and the immune system for host responses during infection is increasingly being recognized. Here we found connections between immune and metabolic genes that are known to drive inflammatory responses. We also found overrepresentation of pathways linked to cholesterol metabolism, suggesting that altered cholesterol mechanisms could exacerbate inflammatory responses in infants with confirmed sepsis. In our metabolomics analyses, we identified 36 metabolic features that were potentially important for separation of infants with confirmed sepsis and no sepsis. And more specifically, based on these metabolic features, majority of infants with sepsis had similar metabolic profiles to each other that were distinct from infants with no sepsis, as can be seen from the heat map. Additionally, we also preemptively identified 5 with biologically relevant metabolite features, including fatty acids and pyruvate, which have been shown to be increased in septic patients in existing studies of immunometabolism during sepsis. So does including multi-omic data improve patient classification? Yes, so when we included demographic data and laboratory data only, we generated 3 clusters. All possible and no sepsis infants were in a single cluster. However, majority of sepsis infants were in one of 2 clusters. When we included transcriptomic and metabolomic data, we had 6 clusters. Again, all infants with possible and no sepsis were in a single cluster. However, now each infant with sepsis were in individual clusters. Of course, this was only a pilot study, but we postulate that integration of multidimensional demographic, laboratory, and multi-omic data has potential applications for stratified medicine. This is a figure from one of my publications, and the main takeaway is that we are currently in this era of empirical medicine, where we are using a one-size-fits-all approach to sepsis diagnosis. However, when we start to include omics data, we could potentially stratify patients into sepsis clinical groups. Eventually, when we include continuous real-time clinical measurements, we could potentially move towards precision medicine, where we personalize the diagnosis and treatment for these septic babies. Ideally, sometime in the future, we would move towards preventive medicine, where we can have predictive biomarkers for sepsis, or we can prevent babies from even being born preterm, since they are most at risk of developing neonatal sepsis. And this leads me to another aspect of my research work, which focuses on the maternal side of the preterm birth equation, where we have used other omics approaches to improve our understanding of the relationship between the vaginal microbiota and preterm birth. So increasingly, the role of microbes, such as in the oral and gut cavities or in the lower reproductive tract, they have been recognized as important in shaping health and pregnancy outcomes. But what do we know about the vaginal microbiota and preterm birth risks? So a lactobacillus dominant vaginal microbiome is characteristic of healthy pregnancy, and specifically, lactobacillus crispatus has largely shown to be beneficial, whereas iners and high diversity type microbiomes have been linked to higher rates of preterm birth. Also, during pregnancy, bacterial vaginosis, or BV for short, is linked to a 2-fold increased risk of preterm birth. Geographical and ethnic differences can also influence the relationship between the vaginal microbiota and preterm birth risk, and that this association is different in Black and white women. Now, despite the fact that most preterm births occur in Asia, majority of existing studies that have looked at the relationship between these factors and preterm birth risk have mostly focused on North American and European populations. Our study was a large-scale characterization of the pregnancy vaginal microbiome and cellulase activity in a low-risk Chinese population. So the main takeaway is that we collected vaginal swabs from 2,796 pregnant Chinese women and performed 16S rRNA sequencing. We then classified 2,689 samples that passed initial criteria into vaginal microbiome groups that were based on the most dominant species in the vaginal microbiome, as shown in the table. We also conducted a sub-cohort analysis in 819 women who had available data on cellulase activity, which is often used as a marker for microbiome dysbiosis in China, and leukocyte wet mount results, which has been used as an indicator for inflammation. As expected, majority of Chinese women in our study were characterized by lactobacillus dominans, and mostly lactobacillus crispatus and iners dominans. The proportion of crispatus and iners dominans in our population were very similar to Chinese and Korean women, so these are the pie charts with the green circles. When compared to non-African Americans and Caucasian women in Europe, the pie charts with the yellow circles, in those populations, the ratio between crispatus and iners dominated microbiomes tended to be larger, with majority being dominated by crispatus. On the contrary, when we look at Asian and Black women in Europe and women in India, as well as African Americans, we find that in these populations, there tended to be larger proportions of iners dominated microbiomes instead of crispatus, as seen by the red circled pie charts, or they had overall low levels of lactobacillus, as seen by the black circled pie chart. So our findings suggest that ethnic and/or geographical differences are a major source of variation in the underlying structure and composition of the vaginal microbiome. In our sub-cohort analyses, we found that high cellulase activity was associated with high diversity vaginal microbiomes that were dominated by BV-type taxa, and specifically taxa such as Gardnerella and Atopobium, which is characteristic of microbiome dysbiosis. When we compared preterm and term births, we found that microbiome dysbiosis, high cellulase activity, and leukocyte presence were not significant drivers of preterm birth in this cohort of Chinese women. So generally, the risk of preterm birth in our cohort was around 5.4%. In the overall population in China, it's around 7%, which is still lower compared to the global average of 10%. So our findings suggest that the lower risk of preterm birth may be due to differences in the vaginal microbiota and or host immune responses of Chinese women. Finally, I would like to reiterate the potential of multi-omics approaches to capture the complexity associated with preterm birth, where we have the possibility to generate new insights that help us to predict and prevent preterm birth, as well as improve the health outcomes for these babies born too early. I would like to acknowledge the participants of our studies, as well as all the people, institutions, and organizations that have made this research possible. Thank you.
Sherrianne, great work. I think this talk and this that you described really illustrates the potential for this omics data and how it can impact us in our daily lives, right? How we can help people around us. So while we wait for some questions to come in, I kind of have a bit of a basic question, I suppose. What's the next step? So it seems like you can identify mothers that might be at risk prior to birth. So does this mean that how these mothers are actually treated during the delivery is changed, or what happens at this point?
Yeah, so there are many, actually, I talked about how there are many causes to preterm birth. So at least for my research, we can look at introducing lactobacillus probiotics to the mothers when they are having their pregnancy, and this could probably prevent them from getting preterm birth. So there are trials currently ongoing, and our group does have studies in that area. So I think that's something that we could potentially apply in the future to try and help mothers and reduce their risk of preterm birth.
Okay, so it's not just about addressing the sepsis that comes with the preterm birth, but actually preventing it itself. Is that correct?
Yeah, so preventing the mom. So basically looking at the mom's vaginal microbiome and trying to make it better by introducing more probiotic type bacteria. And that would result in the mom having less risk of developing infections and reduce the risk of also preterm birth. So then if the babies come out at term, then they themselves have less risk of developing sepsis. So it's sort of like a transitionary effect.
So I guess what I'm wondering is this the only way of kind of preventing that sepsis, by trying to push it to a full-term birth? Or are there other interventions where maybe you can't prevent the preterm birth, but you can address the sepsis that is so often associated with it?
Yeah, so I think it's very difficult because a lot of the times when the babies come out, they have very immature immune systems. So until we can find a way to expedite the maturity of the immune systems, it's very difficult to prevent the sepsis because you need the host response to be mature enough to overcome the infections. So I think in that sense, we are still not there yet, but hopefully one day we could see how we can maybe expedite the maturity of the immune system, or at least prime it to be better at targeting infections.
Yes, hopefully. We have a question from Philip Carlson at Uppsala University. They say, thank you for a very interesting presentation. Do you know how much food choices might affect the vaginal microbiota?
Yeah, actually, I'm not sure what's the answer to that question because I haven't actually looked at the nutritional side of things and how that affects the microbiome. But I think potentially it could affect the microbiome, but I can't say I know the answer to that for sure. But thank you for your question.
So, okay, one more question from Martin Toole. Thank you for the talk. Based on the microbiome analysis and your results, is there anything one can do to minimize the risk of preterm birth in addition to avoiding typical bad habits like smoking?
Yeah, so there are a lot of things that contribute to the risk of preterm birth. I mean, if a mom is overweight, so if they have higher BMI, then that also contributes to higher risk of preterm birth, higher maternal age as well. And if mothers have gestational diabetes, all these factors do affect the risk of preterm birth. So apart from just smoking, there are a lot of other factors that you can try to reduce or prevent yourself from getting in order to increase your chance of having a term delivery.
Interesting. One other really fascinating thing I think I saw on one of the slides is that in the future plans, it's maybe not just testing once to get this this omics data, but maybe serial collection of data throughout the pregnancy. So I'm wondering if you've kind of tried this already, and if you have, if there's ever a time where you see that things are going well, and then quickly goes bad, or vice versa, where you have all these risk factors and then suddenly something changes and they're no longer at the same level of risk.
Yeah, so our group has done a lot of studies on has done a few longitudinal studies where they have tracked the microbiome across different stages of the pregnancy, and they do see that if, let's say, there is a drop of crispatus, then there's a higher risk of the mom having more diversity, and when they have more diversity, it leads to a higher risk of preterm birth. So in that sense, yes, we've done studies on that, and we continue to do studies on that as well.
Cool. We have a question from Leo Chan who says, thank you for the talk. May I ask if symptoms or even resolution of inflammation through breast milk feeding is considered passive immunity, if this is critical for development?
Yeah, so I'm actually not sure how to answer that question because I'm not really in the breast milk field. But I would assume that breast milk would be extremely important for development and it would help to prime the immune system, especially in preterm babies. So obviously we know that breast milk does pass on antibodies. So this would be very helpful for preterm babies as well to give them some sort of boost to their immune system.
Okay. We have a question from Javaria Zafar who says, thanks for the talk. Will there be tailored treatments for every woman since there seems to be varying vaginal microbiomes, at least across international borders, let's say?
Yeah, so I think that would be the ideal place that we can move to. So I think I said in my talk that we hopefully can move towards preventive medicine or even precision medicine. So this is where we start to tailor the treatments and the diagnosis for women or even babies with sepsis. But I don't think we are there yet because there's so much variability and so much that we still don't understand in terms of, host microbiome interactions. So until we understand better, it's very hard to tailor the treatment for each women.
Yeah. And our final question is from Anonymous, who says, more generally, what are some of the known factors or risk factors that influence vaginal biota?
Yeah, so ethnicity does influence the vaginal microbiota. We can see that women with diabetes may also get different microbiotas. So there are a number of factors that can actually impact on whether the mom gets a bad microbiota or not.
Okay, so thank you again, Sherrianne, for this very interesting talk. And thank you to all of the
Thank you.
speakers for participating today in this session. Thank you to all of you for watching and participating so nicely with questions and observing the talks. And we begin again tomorrow at 8 a.m. Singapore time for registered participants. Thank you very much.
Thank you.